Hello, two tenors. We've talked a lot about the benefits of active learning in this course, but you'll see with membrane protein transporters, there are pros and cons to both active and passive transport. We're going to look at the function of this large class of membrane proteins, and these can help regulate what comes in and out of the cell. This really is ubiquitous to all life, and we'll talk about the differences between active and passive transport, as well as primary and secondary active transport. We'll look at some great examples at how figuring out the structure of a protein can help you understand its function. Now, I've included this reference to Lord of the Rings not to refer to your performance in the course. We really do want you to succeed, but rather it's referring to that barrier that membranes set up with regards to allowing certain molecules to pass from one compartment to another. These are the supplemental readings for this lecture. And remember that membranes are lipid bilayers that set up this impermeable barrier, but they can allow for very small, uncharged, or even hydrophobic molecules to pass. You might sometimes hear the word lipophilic, which just means lipid-loving or hydrophobic. Now, if molecules are too lipophilic, they may actually get stuck in that membrane. Passive diffusion doesn't involve any membrane proteins or transporter system. It's simply concentration dependent. So an example would be the waste product that we produce, carbon dioxide, that can diffuse out of cells. And in this case, it's moving down its concentration gradient, and we know that this process isn't saturable. So as you increase the concentration, there would be an increase in the amount going across. There really isn't anything limiting its movement across the lipid bilayer. Transport is really important for getting what cells need in terms of nutrients like amino acids, sugars, and nucleic acids while removing those waste products. So it's really the integral membrane proteins that are going to help move these molecules in and out of the cell. And we would consider this facilitated diffusion, meaning the proteins are helping or facilitating the movement of these molecules across the bilayer. And sometimes they might need a little help in order to undergo the conformational changes that are needed to allow the molecule to pass. If we think about some of the ions or molecules that we talk about in biochemistry, here's an example of things that fly right through, like the gases. While water and urea are right on the edge, they can diffuse across, but not very well. So there are water transporters that can help regulate cell volume. These molecules have to cross the hydrophilic and hydrophobic portions. So when you start to build up to a larger polar structure or a charged ion, they can't get across. Not on here are the proteins, but you should now appreciate just how large they are since they are made up of many amino acids, and they can also be charged. So when you're looking in a cell, proteins really are confined to their organelle or their cellular location. If we left everything to passive diffusion, it would take forever. We need things to move quickly across the membrane for a cell to carry out its function. This is an example of a pore or channel that can mediate transport by facilitated diffusion. Because passive diffusion is quite slow, molecules need to pass first through the hydrophilic head groups and then through the hydrophobic regions. Membrane proteins help by creating a central passage that allows molecules and ions of the appropriate size, charge, and geometry to traverse the membrane in either direction. It's the amino acids that line this channel that will set up an affinity for specific molecules. For example, allowing sodium in but not potassium. You rarely have a situation where pretty much anything is going to get in. The cell just wouldn't like that. So integral membrane proteins are pretty selective for what they move into the cell by facilitated diffusion. Just like what we talked about for passive diffusion, this type of transport does allow things to go downhill, so from a high concentration to a low concentration, or in the thermodynamically favorable direction. The difference between facilitated and passive diffusion is that because we are dependent on a membrane protein to move things into the cell, you can hit a speed limit where transport is saturable. This is just shown here graphically where along the x-axis is the concentration of solutes that you would be moving across the membrane, plotted as a function of the rate of transport. You can see that as we increase the concentration of solutes, passive diffusion increases, but it is quite slow. Integral membrane proteins will speed up this process, but we can see that at very high concentrations, there's a limit to how fast they can transport or a plateau in this rate of transport. We see that it is a hyperbolic curve, so when you get to a point where we're adding more substrate, the transport rate remains the same, and we're going to see that this is similar to what you would see for a simple enzyme catalyzed reaction. If you think about what's going on, there are interactions occurring between the molecule and the protein. So if all the spots are filled up, the protein can't function any faster. It has to wait until that molecule is left before it can interact with another one. A good analogy would probably be entering a TTC station. We don't necessarily have this problem right now. If you're trying to get in through the fare gates, no problem. When we do eventually make it back on campus and everyone's leaving class to head home, if there's hundreds of people trying to get into St. George Station, those gates are going to be quite full and the lineup's going to back up out the door. Now, one way a cell can compensate is to increase the expression of these membrane transporters when they need them the most. We're going to look at enzymes in the next lecture, but what you'll usually see is that they will try to function in this lower part of the curve so that they can adapt and accept more substrate and function much more quickly. So whenever a protein is involved, the rate limiting step really is that interaction. You might have heard about channel proteins before, but these are just a few examples transporting things like glycerol or amino acids or chloride ions, and the structure of these membrane proteins is absolutely critical for their function. Some of the key features are that they are incredibly selective. They can select between a potassium and a chloride ion, which if you know the charge, you can understand, but they can also select between molecules of the same charge. And when it comes to selecting for one versus the other, it all comes down to the size of the molecule. So these channels have very finely tuned filters that determine what comes in and out of the cell. They can also be incredibly quick and can rapidly move ions at the rate of 100 million ions per second. And I don't even think I can envision 100 million of anything moving that fast. The last thing is that they need to be regulated or gated. And this is very important. You don't necessarily want everything to be open at the same time, or you would just hit equilibrium with things flowing in and out, and there would be no way of maintaining ion gradients. So they can be opened or closed depending on what the cell needs. So the cell does have a way of regulating them and turning them on or off. Rod McKinnon was an MD that actually left med school to do research, and this actually resulted in a Nobel Prize. So I know many of you might want to go into medicine, but maybe take this time to really examine what you're passionate about and what you like to learn about, as well as the types of work environments that you would prefer once we do get back to regular society. He was really interested in understanding how ion channels work, since they're involved in a variety of things like regulating cell volume, the secretion of hormones, as well as the firing of neurons and axons, which is driven by these ion channels opening and closing. When he left medicine for research, there was a general understanding of how things open and close, but he couldn't wrap his head around what was going on at the structural level. Peter Agre also worked on a different channel called the aquaporin channel, which regulates the movement of water, and together they were awarded the 2003 Nobel Prize in Chemistry for helping elucidate what's going on at the molecular level and understanding how these channels can select for different ions and move them across the lipid bilayer. Now, Rod McKinnon was working on the potassium channel, and it's composed of 4 identical subunits, so it's a homotetramer, and these subunits are shown in different colors in this side view as well as the top-down view. When the 4 subunits come together, it forms this cone shape when you're looking at it from the side, where there's a larger opening facing the inside of the cell. When you look down from the top, you can see that there is a hole in the middle. This is what an ion would see if it's looking to get across the bilayer. And you can kind of see the relationship of the individual subunits, but it doesn't really tell you how it selects for potassium ions specifically. What they also didn't know at the time is that this is actually the closed conformation of the channel. So remember how I said that these structures need to be regulated or gated? This is actually what it looks like when it's not transporting potassium. This is the ribbon diagram, but if you were to look at the surface representation, you wouldn't see a hole going straight through. The real magic in looking at the structure comes down to understanding the selectivity filter. People had speculated that there's got to be something in the channel that selects for one ion versus another, and it turns out that it wasn't a defined structure, say an alpha helix or a beta strand. It's found in the loops that are contributed by each of the subunits. This is just a cutaway view of 2 of the 4 monomers for simplicity, but each of the monomers contributes a polypeptide loop to narrow that channel and only allow these green potassium ions to bind. The primary amino acid sequence was known at the time, and people had done experiments and made mutations and knew that this sequence was important for activity, TVGYG. Remember, we always write polypeptides in the N to C terminus direction, so it would be threonine, valine, glycine, tyrosine, glycine. It was a bit confusing though, because we couldn't really understand how something like glycine that doesn't have a side chain could be important for binding to these ions. You would expect to maybe find some negatively charged amino acids there playing a role in filtration. It turns out that it's not the side chains but the backbone carbonyls, so the C double bond O's, that are pointing in along with threonine's hydroxyl, which is at the bottom of the filter. I hope you can appreciate just how novel this finding was. No one expected the backbone to play a role, and it does come down to the geometry and the chemistry at play allowing these functional groups to interact with the potassium ion. The other thing to keep in mind is that the potassium ion actually becomes partially dehydrated. So because it's charged, it's very soluble in water and likes to interact with water outside of the membrane transporter. But for it to be transported, it needs to release its shell of water so that it can fit in the pore. So if you were to look at the geometry of binding, the carbonyls are actually replacing the bound water molecules as potassium enters the channel. There still will be a water molecule between the potassium ions that are being transported. Remember, this is in 3D. But the waters that would be on the sides are going to be replaced by carbonyls. This also makes sense because there will be electrostatic repulsion between the potassium ions, so you would see a water molecule both above and below the potassium as it's traveling through the channel. Now, if you change the sequence of this reentrant loop, you can actually change the ion selectivity of this cation channel. We can see the conserved glycine residues in pink, as well as some other amino acid changes within the sequence. Changes to the side chains that don't interact directly with potassium can affect the overall structure of the peptide loop, but it's the size of the side chains that affects the orientation of the backbone carbonyls. So they're still contributing to the structure of the reentrant loop, but not necessarily having a direct interaction with that potassium ion, with the exception being that threonine side chain, which can also be mutated. So this stretch of 5 amino acids is forming the scaffold for transport and acts as the selectivity filter. This slide is just showing you the major conformational changes that occur between the closed state that we just looked at and the open state of the potassium channel. Years later, Rod McKinnon did publish the structure of the open state, and it's not as nice. But what we do see is that the selectivity filter is still intact when it's open. It doesn't collapse. So these red loops don't move significantly, and they still are able to ensure that only potassium is transported across the membrane. What does happen is that the base is wide open and allows ions to freely traverse the bilayer. Now, there's a key glycine residue, glycine 99. Remember, glycine is the smallest amino acid, and it's found in one of the 2 helices. So this allows the bottom part of the helix to hinge or move out of the way. In the case of the potassium channel, the gating stimulus is either a change in voltage or pH. So there's a sensor in the channel or amino acid side chains that can sense these changes and allow for the conformational changes to occur and open the channel. We've talked a lot about the structure of membrane proteins, and for the most part they've been alpha helical, but not all membrane proteins have this structure. You can also see beta barrels that consist of many beta strands, and remember, this is the type of domain that'll form a pore that allows molecules to cross the membrane. These membrane proteins are a little different in that they don't undergo major structural changes. When they're present, for instance, porins that are found in the outer membrane of bacteria, they are less selective and can let hydrophilic molecules like glucose into the cell. So they really aren't as stringent as the potassium channel that we just looked at. An important point is that there are similar patterns in their amino acid sequence. So remember that beta strands have a more elongated structure, and for a beta barrel to form, you would have an alternating pattern of 1 hydrophobic and 1 hydrophilic amino acid, so that every other side chain would be hydrophilic and pointing into the aqueous middle of the barrel. So if you see a sequence of alternating hydrophobic and hydrophilic amino acids in the primary sequence of a membrane protein, you could maybe predict that it would be found in a beta barrel. Active transport is the movement of molecules against their concentration gradient. In these images, you can see that the membrane protein needs either the input of energy, so from ATP or light or the passing of electrons, in order to undergo the conformational change needed to move an ion across its concentration gradient. These are sometimes referred to as pumps, and you can see that they do interconvert between two conformational states. We've already seen a few examples of primary active transport in this course, but it'll also come up in the metabolism section when we look at the electron transport chain. Secondary active transport basically just uses the concentration gradient of one molecule to move another. So you'll have two different binding sites for the co-transport of two molecules. For instance, A could be going down its concentration gradient, and this would result in a conformational change in the membrane protein that allows for B to go up its concentration gradient. And transport can either be in the opposite direction, say for an antiporter, or in the same direction for a symporter. There are two different ways ATP can help mediate this change in a membrane protein. When ATP is hydrolyzed, free energy is released that can be used to change the structure of the membrane protein and allow for the transport of a molecule. We already saw that CFTR can bind to ATP, and this part of the protein is sometimes known as the nucleotide binding domain, where hydrolysis of ATP to ADP and PI allows for the conformational change needed to transport chloride ions. You'll sometimes hear these referred to as ABC transporters. Multidrug resistant proteins also use this mechanism to export drugs out of the cell. The other way ATP can drive active transport is by donating a phosphate to phosphorylate amino acid side chains, and this will result in the conformational change. These membrane proteins may also be known as P-type ATPases. Here's just an example of a flipase where ATP hydrolysis is helping to change the conformation of the structure, allowing for the movement of a phospholipid from one side of the membrane to the other. On the left is the open form where ATP isn't bound. And then once ATP does bind, this is what's helping drive or change that conformation in the membrane protein structure, allowing for the lipids to be transported from one face to the other. The last example I want to discuss is Bacteria-rhodopsin, which we've already saw has 7 transmembrane segments. It also has a retinol cofactor that not only contributes a purple color, but also can undergo a conformational change in the presence of light. This is an example of a light-induced transporter that transports protons up their concentration gradient. Bacteria would then use that proton gradient to generate ATP, and ATP synthase would also be an example of facilitated diffusion. So we saw Bacteria-rhodopsin in the last lecture where we analyzed the amino acids present using a hydropathy plot, and we saw that there are 7 TMs represented by the letters A to G. There are also amino acid side chains found in these helices, and these can help bind to protons and transport them across the membrane. Lysine 216 is also covalently attached to the retinol cofactor, and when it absorbs light, we can see that it is a chromophore, so it has lots of double bonds, and you actually get this huge change in structure that will affect the nearby amino acids, and this will result in their protonation or deprotonation. So remember when we talked about how pKa values can have different numbers depending on their environment? This will allow different functional groups to either be protonated or deprotonated, allowing for protons to be pumped across the membrane. Now, this figure only shows 2 acidic amino acids, but there are other amino acid side chains that help with proton pumping. What's really neat about looking at Bacteria-rhodopsin's structure and mechanism of action is that the protons don't diffuse across the membrane. They essentially hop or are passed from one side chain to another. This is similar to what you would see with free water, where as one hydronium ion gives up a proton, the one adjacent to it binds and accepts it. True diffusion of the proton across the pump would mean that the proton that enters the membrane is the same one that is exported immediately if you were to track its transport. Bacteria-rhodopsin uses proton hopping to pass a chain of protons through different amino acid side chains. So as soon as one's picked up from one side of the membrane, another one is immediately released. The proton will eventually exit the transporter, but it's through this series of hops along the different functional groups that allows transport to occur quite rapidly. So the structure of membrane proteins really is important for understanding their function. We see that conformational changes are key for the movement of ions and molecules across the membrane and can also help with the regulation of transport. Some membrane proteins, like the beta-barrel proteins, can allow multiple molecules to cross the membrane, while other membrane protein transporters are more specific. Transport can either be passive or active, and active transport uses energy or concentration gradients to move molecules across the lipid bilayer up their concentration gradient. Keep in mind that transport is also saturable, so this is all due to the interaction of the molecules with the amino acid side chains found in the proteins. Check out the 2 brain break questions before moving on to the last lecture for the week.